Thank you so much, Mindy, for the invitation, and thank you everyone who came to attend to today's wonderful program. I think it's a great idea to educate about Parkinson's disease. I love doing it. We also have a bunch of programs. Many of them are hybrid, just like yours, and there's never enough learning or too much learning, so I will share some of the upcoming programs at the end of my talk for you to potentially sign up for. All of this is free, again, available in a hybrid format. You are welcome to come down to Las Vegas, and we have some nice food there. We are proud of our hospitality, as well as you are welcome to attend remotely. We don't have any limitations or costs involved with that. So today I would like to give you a sort of a pretty large chunk of where we stand with Parkinson's disease research. It will touch on a number of specific projects, and it is a lot of material. So what I usually do is put together a number of slides and material that is way too much for the allotted time, and I stop here and there explaining something that I find maybe very important or relevant to you, and skip other slides as I go along. But I'm very happy to share the slides with anyone. I created this presentation. There's nothing copyright protected about it. So I put one of the slide copies on this computer here, and I'm happy to have you or anyone to get the full access. I'm just saying that because there will be slides that I will skip for the sake of time, and hopefully we will have some time in the end that we can use to discuss questions that you have, and I still make it to my flight. Okay, so I have many disclosures. I'm not going to read them. I do consulting for fee and advisory boards and all that. Anyone in movement disorders of some relevance would have that. I have also some other conflicts with regards to these professional organizations where I have some roles, and they are all listed here. Okay, so first of all, I think the part of it will be blocked on the top, but what that says there is outline. I would like to go through a number of research studies and ongoing trials and other information. However, what I would like to do before I do that is spend a number of slides on explaining what research is. What we mean by research, and what kinds of research do we have to know about in Parkinson's disease. So you will have a little bit of an idea about those. So this is just the definition of research: a systematic investigation into the study and materials and sources in order to establish facts and reach new conclusions. So that's the Oxford Languages definition of what research is. And of course, research relevant to Parkinson's disease will include some very basic research where we study a molecule and understand potential biomarker properties. That is entirely biochemistry in the lab. There's also animal research. All of that is preclinical, where there's nothing about humans at that point. And there's also translational research where there is something human about it. There's a biomarker that's being tested. There's a human tissue involved, and all the basic science knowledge is being sort of interfaced or correlated with ultimately human trials. And then human subject research is what would be the next level after that, where we would understand a biomarker, a diagnostic biomarker, or progression biomarker, or other things, or try some intervention in clinical trials to see if we can advance treatment by that. So what is basic science research? The basic science research, like I said, is something that happens before anything reaches the realm of human testing or human experiments or research. And at this level, it is something that contributes to the fundamental knowledge that is the basis of any further steps. Because we get a lot of questions like, why waste all that money into something that doesn't even get out of the cell, all that attention to molecular mechanisms? And there's a good reason for that, because without that understanding and knowledge, we would not have much to do at later stages of research. And before I get into, so that was the definitions of some of the basics of what research is. I will have some more slides to go through some specific types of research, what is phase 1 and phase 2, etc. But before I do that, I wanted to share that my best source of research updates in general is my friend Mike Okun, who is one of the most recognized movement disorder experts in my field. And he was a friend. He was a few years ahead of me in fellowship. We did that at Emory, and I was at NIH. And as Dr. Lakshin mentioned, my first job was at Johns Hopkins, and they asked me to build a DBS center. And then in the early mid-2000s, that was a new thing. DBS was relatively new at the time. And so I looked at Dr. Okun, Mike, as a friend, because he was a few years ahead of me, and he just created a multidisciplinary DBS center, of the first of its kind, a few years before my time, in Gainesville, Florida. And it still is the most advanced and most comprehensive DBS center in the world. And so the whole notion of how we do candidacy determinations, everything came from him when I was just starting my career. So I'm very grateful for him for that, and many other ways that he helped me throughout my career. But anyway, for research updates, I use his Twitter and Facebook social media. If you are on social media, find him and sign up to receive those updates, because I believe that that's the best way to keep up, because he always comments on anything and everything that is of relevance, that just got published in any one of the journals, and he filters it for you and also adds a very useful commentary so that you can kind of put it into perspective as to why this is relevant and what it means. This is good for doctors like myself and movement disorder specialists. As you can imagine, some of the information could be a little bit too technical for somebody who doesn't have a medical background, but nevertheless, this is a high-end consumer level narrative. So I believe the majority of you who come to these talks and spend time and effort in educating yourself about Parkinson's disease will understand much of it, even though the referenced peer-reviewed paper that he is commenting about may be a little bit too technical for non-scientists. His narrative will not be too technical. So again, the basic research is something that I already touched on, and I will not talk more about it other than repeating that without the fundamentals that only basic science and basic research can produce, no other research or science would exist. So the difference between applied research and basic science research, to make it simple, is essentially that the basic science research has no purpose whatsoever other than generating new knowledge, understanding something that we don't understand, creating some kind of information and understanding for the purpose of just that understanding. Whether that will translate into something valuable, applicable, clinically useful, ever, is not their concern. They just want to go after problems that exist and knowledge that doesn't exist and resolve those issues. That's the only purpose of basic science. And then, again, like I mentioned earlier, there's a lot of criticism as to why waste all that money for something that doesn't lead to an application or doesn't lead to the cure. And it's a good question. And it's not that easy to understand that the way that science works, if we don't have the fundamentals, if we don't understand the very basics of how nature is functioning, if we don't have those very small and very primitive and very non-practical blocks of knowledge, then anything that would lead to applications would be missing. Of course, applied research has a purpose. It does have, like for instance, a lot of pharmaceuticals and medical device companies will have a quote-unquote basic science realm and internal research teams, and so they are supposed to do basic science, but they are not. They are paid and they are asked to develop something that will surely help their pharmaceutical or medical device development realm. And universities, and only universities who are only interested in understanding nature and for the sake of understanding, will generate that very critical fundamental part of knowledge that everything else will depend on. And of course, there are grants from industrial and pharma in the private sphere to support basic research, but nothing can completely conflict-free fund basic science research other than the public itself. So I already covered this to some extent, so just in a couple of additional bullet points. Basic research seeks generalization, attempts to explain why things happen, and this foundation is going to serve as a basis for applied research. And applied research, as the name suggests, has a purpose besides just generating knowledge. It has a purpose of an application that will be useful for making money or making our lives more efficient, reducing mortality, or whatever it is. By nature, applied research is certainly more specific and again has a practical purpose. There's another way you can classify research, and that is quantitative and qualitative research, and then there's also mixed research, and both are important. To some extent, each will contribute to our understanding. And again, I'm not going to read out every bullet point on every slide. I just wanted to emphasize and single out a few important aspects of it. So, much of what I said about applied research, basic research, qualitative research, quantitative research, that kind of is true for any research, in biology or chemistry, anywhere. And now we are now turning our attention to medical-related research. It's sometimes referred to as biomedical. And so the kind of, and I'm sorry that some of my headings will be cut off here, but I wanted to go through a few specific types of research terms and what they mean within the medical research realm. So basic science research, bench research, we already covered that quite well. There is such a form as case series or case reports. a lot of journals and generally the medical academic world looks down at case reports, as anyone can do that. It's easy to do, and it doesn't add a lot of knowledge. There is some truth to that sort of negative assessment. It's not a very valuable contribution, but there are certainly great cases that can help us learn something, and it is still remains a very important part of, if you look at a clinical journal, there will be case reports and case series, quite a few. Observational natural history studies, and I will specifically single out a couple of those as we go along. There are two that we are enrolling for at our center as well. Observational studies are not interventional. They are not trying to prove a treatment to be effective or ineffective or safe or any of that. What they are trying to do is understanding the disease better, what it is doing, what subtypes may exist, how they behave, and that understanding and classification will further our knowledge as to how potentially different treatments can exist, how we can advise patients about prognosis. For instance, in Parkinson's disease, there are patients who have tremor, and then we refer that as a tremor predominant form of Parkinson's disease. There are also patients who don't have tremor and starts falling as one of their early symptoms of Parkinson's disease, sometimes referred to as PIGD phenotype. The PIGD stands for postural instability and gait disorder, and it is from these natural history studies and observational studies that we know that people who have the tremor predominant variant of Parkinson's disease do better. The rate of prognosis tends to be slower. And there are many other such knowledge that can help us advise patients in the clinic, and also can help us design better studies when we know how certain disease types or diseases behave over time. So understanding the disease better with these natural history protocols is a very important piece of our knowledge base. And also, I just wanted to emphasize that when we are enrolling patients in these observational studies, and they say, "Well, nothing is being tried. Am I contributing in any which way?" The answer is absolutely. Without that knowledge, we wouldn't have biomarkers, for instance. And a lot of major breakthroughs, especially recently, actually came from natural history and observational studies. Next up is chart review and retrospective analysis. Again, oftentimes looked down as a relatively low quality type of research. And the reason for that is that people feel like when we are looking back into the past, we're not controlling our hypothesis. We cannot really find a good control to our research questions. It could be biased. I only look at the charts that want to prove the point that I'm doing the research for. So there are a lot of flaws and problems with chart reviews and retrospective analysis. Having said that, there are completely valid and valuable retrospective analysis. One is case control study. We were just talking about one with Dr. Lakshan earlier, where Medicare database analyses have a research question, and before you start sampling your database or accessing it in any which way, you have a research question that you write up front, so you have a hypothesis and you have a design for pursuing that hypothesis testing by setting up proper controls even before any chart was opened. And so once you have created that study design, one that I was mentioning to Dr. Lakshan earlier was where they looked at whether GLP-1 analogs, and I will talk about that very interesting topic in a little while. The GLP-1 analogs are very popular, like Wegovy, Ozempic, Ozempic, Zepbound, Mounjaro, all of these originally anti-diabetic drugs that are repurposed for weight loss now are being looked at as potentially slowing down neurodegeneration, including Alzheimer's and Parkinson's and other forms of neurodegenerative disorders, chronic traumatic brain injury, as well as even normal aging. And one way that people wanted to look whether they make any difference is setting up a question which was, incidentally, will Parkinson's disease become less likely if somebody's on these drugs versus a control population? So they have set up perfectly what the control should be, age matched, sex matched, and any other demographic and comorbidity profile should be matched. And so the algorithm was searching for individuals who are on these drugs versus individuals who are not on these drugs but otherwise perfectly match the target cohort. So there's a control cohort, there's a target cohort, and they were matched on age, comorbidities, their other medical profile, their other medications, their diagnosis, everything. With one difference: one would be on one of these GLP-1 analog drugs, and the controls would not be on that drug. And anyway, there were thousands and thousands of cases, both in the study population and in the control population or control cohort. And interestingly, what these analyses have been pretty consistently finding is that the incident Parkinson's disease rate is markedly reduced in those populations who were on these GLP-1 analogs. But you know, it doesn't necessarily mean automatically that it will cure Parkinson's or slow the progression. And the naysayers point out to some of the negative studies that looked at the slowing effect of these GLP-1 analogs, and they have so far came up pretty empty-handed, because affecting the risk of Parkinson's disease, which is what the incidence rate over time looks at, versus affecting the rate of progression once you do have Parkinson's disease, are two different questions. And one example as to how those two things are different and sometimes separable, and you cannot equate those two things, is the LRRK2 story. I'm sure many of you heard that there is a leucine-rich kinase called LRRK2, or LRRK2, and that's one of the most common genetic conditions that increases the risk of Parkinson's disease. You know, no mutation will cause Parkinson's disease in a sense that everybody who has that mutation will have Parkinson's disease. That doesn't exist. But there are many, many so-called risk genes in which if you have a known mutation, then your possibility of developing Parkinson's disease sometimes in life will be increased. How much increased? It varies on the Parkinson risk gene. Some of them have a higher rate of risk increase, others increase the risk minimally. So one of the most common so-called Parkinson risk genes is the LRRK2 gene, and the LRRK2 gene produces the LRRK2 enzyme, and this mutation that elevates the risk of Parkinson's disease is a so-called gain of function mutation, meaning if there is a problem, if you have the mutation, and so there is an error in that gene, it expresses more of the product, which is the LRRK2 enzyme, than less. And so in that regard, if the LRRK2 enzyme is higher, at a higher level than normal, then the risk of Parkinson's disease will be greater. There are many different mutations in the LRRK2 gene, and they are not created equal. Some of these mutations they are all gain of function, so meaning every one of these Parkinson risk-related mutation in the LRRK2 gene will elevate the risk of Parkinson's disease. Some of those mutations will increase the LRRK2 enzyme production a little bit over the normal, some more, and some others even more so. and they looked at how will that correlate with the risk of Parkinson's disease. And there's a dose-response curve, meaning the more LRRK2 enzyme you have, the greater the gain of function, overexpression of the mutation, particular mutation is, the greater the risk of Parkinson's disease. So that looks like a very relevant finding, and therefore, obviously, we can affect the risk of Parkinson's disease in individuals who don't yet have Parkinson's disease, but have a LRRK2 mutation by taking out the excess LRRK2 enzyme, and there is a treatment for that. It's not commercially available, but it has been established, and the company that owns that investigational product is called Neuron 23. And so one thing that can be done right now is to individuals, and it's happening, individuals who have that mutation will be on this treatment that reduces their LRRK enzymes, and whether or not they would have developed Parkinson's disease or not, they would have an increased risk for that. And they can get a treatment that will normalize that risk. It will not make the risk go away, but the lifetime risk of Parkinson's disease in a general population is probably between 2 and 3%. So if you have the LRRK2 mutation, some of them will increase that to 40%. And that means that you still have 60% of not ever having it. So it's a judgment call whether a treatment that can cause side effects and could be detrimental in other ways is worthwhile taking just to reduce that risk. But what they are also doing is looking at how perhaps the same product that reduces the LRRK2 enzyme will slow down the disease progression. So anyway, where I came from is the GLP-1 analogs and their effect on Parkinson's disease incidence could be studied in a well-designed and meaningful retrospective analysis in that large Medicare database analysis. There are other types of research studies. I'm not going to call all of them out, but we will talk about clinical trials. And so many of you know clinical trials are generally classified as phase 1, 2, 3, and 4. In this particular slide, I have the preclinical, that is the basic science, the translational science, all the things that I was talking about in the beginning. And then once, and that includes the animal testing as well. And the animal testing itself is taking quite a bit of time, and usually they use several different animals and species before anything is allowed to go into humans. And so as far as what kind of animals we can use and pick, there's a lot of thought that goes into that, because animals are not created equal. First of all, no animal will naturally have Parkinson's disease. Only human, Homo sapiens, is the only species that is known to have Parkinson's disease. So when it comes to Parkinson's disease and we pick animal models for Parkinson's disease, they're not going to be perfect. They will have some form of unnatural or not perfect representation of the human disease. And how we make those choices and because there's got to be some compromise, but we do have at least a choice as to what compromise we take. And I could talk a lot about animal modeling if we had time. Maybe another time I just come up and talk about just that. But anyway, as far as the phase 1, 2, 3, 4, I wanted to spend just a couple of minutes to make sure that we cover that. So the phase 1 is the first in human step, where something that has not been tried in humans before now is being tried for the first time. So almost always it's done on healthy, normal controls, individuals who are paid to be the guinea pigs of something that has absolutely no previous knowledge of what it does in humans. And there are very strict and careful protocols involving dose escalation, where a tiny amount of this new substance investigational product that has never been tried in humans before is given, and then they observe, they look at the person. There's a very detailed analysis of their blood, their brain, their spinal fluid, their function, their imaging, etc., and depending on what the actual compound may be. And then the dose gets to the next level and the next level after that. And so from the animal studies, there is a pretty solid understanding of toxicity, what levels were found to be safe, and also efficacy, because all the animal studies are not just safety studies, they are looking for efficacy, whatever the model allows, because that's kind of a very big question. But the bottom line is that there is a generally pretty good idea when it gets to humans as to what the dose range should be that is likely effective and safe. And so the safety has to be established. There is absolutely no efficacy typically looked at in phase 1. That's why it's normal volunteers, healthy controls, not the target disease. Very rarely in truly spectacular or breakthrough treatments, where it's like new in class, something that hasn't happened before, it's not unheard of that even phase 1 is done in patients, and the rationale for that and why IRBs would be amenable to approve that is because this is such a tremendously game-changing thing that you want to allow patients to participate in as early as something can be tested in humans. It is typical for deadly diseases like really aggressive cancers like glioblastoma and other things, where patients have a death sentence and they are dying very quickly, and now there's a new treatment that can change that. They will allow phase 1 testing for such investigational products in the target patient group. Otherwise, it's done in healthy controls. Phase 2 is the next. So let's say a compound gets through phase 1 testing and it's found to be safe. The next level up is phase 2. Now, phase 2 studies are done in the target disease population. So in this case, Parkinson's. And phase 2 studies are generally much bigger than phase 1. Usually in the hundreds is what the goal is. And the FDA has some level of flexibility as to what they require for an approval, for a NDA, a new drug approval. So once you have a phase 2 that enrolls hundreds of patients, and it is extremely safe. So most phase 2 studies are still powered and geared towards safety outcome measures. So that's still the primary goal of phase 2. But they also include efficacy outcomes. So like I said, phase 1 doesn't have any efficacy outcome whatsoever. There's no endpoint for that. And then phase 2 will have some efficacy outcomes, but the primary outcome is still safety. Now, there are pretty robust efficacy outcomes in many cases, and the FDA, for something that is a breakthrough therapy and has the expedited pathway, will agree to accept two separately done phase 2s by different investigators, usually a slightly different study design. And if they are both robust and it's a safe drug, and it is a very relevant drug in the disease, it's a big game changer, they will not force the sponsor, which is the pharma company, to go through the phase 3, which is a lot more expensive and time-consuming phase, and accept and approve the drug based on two separately done phase 2 studies. Now, phase 3 is the gold standard before approval, and that's pretty much similar in any other jurisdiction and country around the world. Phase 3 are necessarily multi-center, meaning it is not just one site that enrolls patients for this, but there are or multiple. They are usually looking for multiple 100 to 1,000 level subjects to be enrolled. They are always placebo-controlled, and they have the primary endpoint as built around efficacy. So, and again, that's the gold standard, and most FDA approvals are done based on phase 3 studies. So I'm going to skip many of the other research slides, except to say that when it comes to clinical trials, the distinction I wanted to emphasize for a moment is what differentiates symptomatic versus disease-modifying clinical trials. And that's an important distinction. I have been mostly interested in most of my career in the disease-modifying realm, and I have done dozens of clinical trials. The majority were disease-modifying. And what symptomatic and disease-modifying is an important distinction. So right now, all the treatments we have, except for exercise, because exercise is medicine, but outside of exercise, every treatment we have, including medications, surgery, interventions, focused ultrasound, PT, OT, speech therapy, all the treatment modalities and treatments that are currently approved and available for Parkinson's disease are symptomatic. They aim to reduce the impact of one symptom or another, meaning they don't treat the disease, they don't make the underlying pathology go away, or the progression rate slow or stop. They just make the symptoms that are disabling less biting, easier to live with. It's almost that you will swipe all the problems under the rug so you don't have to deal with them, you don't see it. It's not in any way knocking it down. I'm not trying to diminish the significance of symptomatic therapies, because they are game changers. They made a tremendous difference in Parkinson's disease before the time of levodopa up to the 1960s. It was an absolutely devastating disease where patients became bedbound in a few years' time, and they just died because there was nothing to do to help them. And so with the advent of symptomatic treatments, we made a deadly disease, a chronic condition, which is livable for decades, and depending on the age of onset, it barely affects the lifespan or life expectancy. The health span is affected significantly, but nevertheless, it is the marvel of these symptomatic treatments of how much of a difference, if properly used, they can make, especially in the motor symptoms. Maybe not as much in the non-motor domain, but those are the symptomatic treatments. The disease-modifying treatments, on the other hand, which we don't have, minus exercise, because exercise is slowing down progression, but none of the treatments that we have tried to slow down or halt or prevent future progression worked. So we are still trying and still working on it, and we have not done very well, because all of the ones that have been tried so far, none of them actually produced anything good. And the question is, why is that? Well, that is an important question, and a lot of us are working on trying to figure it out and maybe fixing some of these barriers that cause the zero translation. When I showed you that big graveyard of various compounds that were once celebrated as the new cure, or disease-modifying neuroprotective treatment in Parkinson's disease, the celebration came from very, very positive, very promising preclinical studies and those animal experiments. I mentioned to you that none of the animal models are good or good enough. They are imperfect, and the imperfection may be translating into this complete lack of success of disease-modifying interventions that are not seem to be working in humans, even after they worked perfectly in animals. And so we are still at that stage. And the bottom line is that we are not in a complete darkness about this. We do have some clear points and contributors and understanding as to why that may be. Why is no translation between animal model successes, glaring phenomenal successes, into complete zero success in humans? And so our molecular understanding of Parkinson's disease has a lot to do with it. There are other factors here that I listed, but I think where we can make the biggest difference is twofold. One, molecular understanding of Parkinson's disease, a better understanding as to what exactly is causing the pathology, what are the molecular pathology pathways and underlying etiopathogenetic facts that eventually are responsible for the neuronal loss and the neurodegeneration. If we can piece that together, including a great degree of heterogeneity, every patient is different in this regard. We just need to characterize them better, understanding their individual etiopathogenesis and pathology better in order to target it and develop better targets. So that's happening, and those observational studies that I mentioned earlier are critical in that improved understanding of the molecular pathology. How is synucleinopathy linked to neurodegeneration? How are these genes that I mentioned that increase the risk of Parkinson's disease and their products at a molecular level, how are they contributing to disease? So I don't want to just create another talk on the causes of Parkinson's disease, enough to say that it's variable individually and it is not a single thing. It is always a combination of factors. When I mentioned the risk genes, I told you that there is no causing gene. There is, like in Huntington's disease, there is a causing effect of the genetic mutation. If you have the mutation, you will have Huntington's disease. Everybody, at a certain level of CAG repeats, there is just a question of time. If you live long enough, you will have 100% Huntington's disease. There is no such thing in Parkinson's disease. The highest risk increasing genetic mutations are still not 100%. Even if you have GBA, LRRK2, any of the Parkin mutations, any one of these genetic mutations, you could have the mutation and live 100 years without Parkinson's disease. So that alone is not going to explain. So what I'm trying to say is that it's always multifactorial and multi-etiologic, where you may have some genetic mutations, or you have just a particular genotype that you bring in, and then you are exposed to some kind of environmental factors, pesticides, herbicides, Agent Orange, the list goes on, TCA. And once you do, then the combination of these things will start something. I also have a research featured which talks about the gut-brain interaction. So the bottom line is that we can affect the lack of translation from the basic science and animal modeling successes and sort of lack of clinical trial successes by understanding our targets better, what we should target more, by our elevated biochemical and molecular pathology understanding of the disease. And number two, going earlier. Because that's the other part. Once a process starts, we believe it's a cascading event, and once the horses are out of the barn, which is one of the explanations as to why we have this lack of translation, so once the process starts, it maintains itself. So we have to go to the inception. We have to go back in time. Now, with animal models, you can injure the animal or inflict any sort of change that will eventually lead to the Parkinson-like process, and then use the prevention before that time point. In humans, up to recently, we didn't have a time machine to go back in time before the Parkinson started and try to do a preventative intervention at that time. What is different now is we have the understanding of prodromal conditions. Prodromes or prodromal conditions are changes that are associated with Parkinson's risk later in life. So we know if somebody loses the sense of smell and there is no other explanation, it is referred to as idiopathic anosmia. I'm emphasizing the idiopathic, which means no known cause, because you could lose the sense of smell for a number of other reasons that are known causes. If you had COVID and you lost your sense of smell, that's a common cause. If you have a skull base fracture that will share all the olfactory fibers, you will lose a sense of smell, but we know why. But if we don't have any explanation, we don't have any other reason, you just lose your sense of smell, you are up to 80% likely to have Parkinson's disease later in life. And that 80% is greater than any of the risk genes that I was talking about earlier. Same thing goes with RBD, REM behavior disorder. If somebody has idiopathic RBD, meaning that they act out their dreams in the REM phase, they similarly 70 to 80% risk of Parkinson's disease later in life, and we don't know what to do, because we know that you are on track of developing Parkinson's disease if you have anosmia or RBD, but that creates an opportunity for these disease-modifying treatments to be tested. It is, if you will, it's our way of a time machine, because these prodromal conditions will tell us that those individuals have a very high risk of developing Parkinson's disease at some point, sometimes years later, and now we can exercise the intervention at an earlier stage in the disease. So that's something that is very important. This paper I wrote with Dr. Mestre on this very problem, as to what is it that prevents us. I kind of have both sides in my experience. I was working with animal models earlier in my career, and now I'm a clinical trialist, and so I have a particular experience in and frustration as to why we can't get this done. I am going to go to another gear so that we have more time. By the way, this is an experiment we did at Hopkins along those GLP-1 analogs, where we try to understand how it works. So we had a couple of, these are the best animal models that exist, the transgenic model, as well as the PFF model. So the transgenic model basically includes the human mutated alpha-synuclein that is injected to a mice, and so the mice brain will express the abnormal synuclein and starts a Parkinson-like process. And the PFF model is that we inject misfolded alpha-synuclein, the bad form of alpha-synuclein, into rat brain, and it starts propagating and turning every normal wild-type alpha-synuclein in the brain into bad one. And so in these two models, we were able to explain as to how GLP-1 analogs stop and prevent the pathology. Again, unfortunately, so far it didn't translate into a successful clinical trial yet, but it's being done still. And I wanted to also highlight the gene therapy approach that is being done right now, and I'm so sorry that it cuts off all the top, but this is a experimental compound which uses an adenovirus-based viral vector injecting glial-derived nerve growth factor into the brain. What the way that the gene therapy works is that this particular gene that expresses the GDNF is inserted into cells in the striatum, and so they start expressing GDNF. GDNF, or glial-derived nerve growth factor, is a protective agent. The growth factor makes neurons survive better and die less, and we believe that with the slow progression of Parkinson's disease, what's happening is that a lot of neurons get hit by the neurodegenerative process. but they don't die right away. They get into this senescent, half dead state where they don't function, but they are not dead. And with the growth factor, they can be reignited and brought back to function. And so the hope for this study is not just that the growth factor will protect the living neurons and prevent the dying and slow down their dying from the neurodegeneration, but it actually could even go a step further, which is that neurons that are not functioning could come back in a way that's a restoration, not just neuroprotection. So that's a very exciting study, and we are enrolling for this down in South. I wanted to mention that not all hope is lost on the GLP-1 analogs. There was one study by Dr. Foltine in the UK that showed some effect, so there is more to be done. There was another study in Europe that showed the effect of another GLP-1 analog. So there are some negative trials and some positive ones. This is the Medicare database search that I was talking about. You look at the numbers. The comparable anti-diabetic is DPP4I. Those are the individuals, 51,000. So that for a chart review, that's a pretty impressive number. So as you can see, these are the two cohorts that I described earlier. And again, they identified 89,000 Medicare beneficiaries to start. They all had diabetes. They were all on diabetic medication. And some of them were on GLP-1 analog. Some of them were not. And look at the incident Parkinson's disease rate. It's very divergent. So it does protect. It does make Parkinson's disease much less likely to happen in the population that's on this. And by the way, we talked about this with Dr. Lokshin earlier. When we look at these diabetic purposed use of GLP-1 analogs, they don't get into the brain very much. They are not purposed to get into the brain. A little bit will get through the blood-brain barrier, but not a whole lot. And even as little as it makes it through the blood-brain barrier creates this humongous effect. So that's very remarkable. And this is the other study that I mentioned that was positive. This is the previous one with Dr. Foltine was exenatide. This is lixisenatide, and that was a multi-center trial. The one with Dr. Foltine was a single-center study. This is a multi-center study. Okay, so I think my time is up, and like I said, we have a lot of other studies that I featured here. I put it into the, and by the way, this is a pipeline series. So Dr. Farting and the colleagues, each year, I highly recommend this paper. I was hoping it would come out because it comes out always in the first week of July. This year, for some reason, the 2025 edition is not there, but there's a 2023, 2022, and this does a fantastic job of cataloging every clinical trial in Parkinson's disease in the disease-modifying and symptomatic arena. The only thing it doesn't do is doesn't do procedures like DBS or focused ultrasound. All those kind of things are not included in this review, but it's an extremely helpful, and the 2025 update is I would say any minute now. And so that's how they show you all the different symptomatic and disease modifying, which are phase 1, phase 2, phase 3, each individual agent, or rather the mechanism of action, what they are targeting is color-coded. So it's a very, very nice review of everything that's happening out there. So if you just remember two things from this talk, get the paper from Dr. Farthing and others, and get your updates from Mike Okun's social media. You're gonna be set, because these keep coming. So, you know, the whole point of Research Update is that things are changing. And so these are both very good in that. And again, I will not spend more time, just to mention a couple of studies, I mentioned the AskBio one with the adeno-associated virus gene therapy. PD gene is a natural history study that we are enrolling at our site, and you get not only right away you get a free 7 gene, the most common risk genes in Parkinson's disease. You get the results for free for participation, but now they are adding whole exome analysis as well. So any other gene relevant besides those most common 7 will also be included. We are also just activated for the next phase of the phase 3 of the prasinezumab, which is a monoclonal antibody against the bad synuclein. We have that at our site. I just wanted to say that we are we are saddened by the federal support for Parkinson's research being rolled back. That's not the way to go. I know it's partially a political issue. This will disrupt a lot of ongoing research studies, unfortunately. And we have a program that you are welcome to come. I mentioned in the beginning that we do a lot of educational programs, and this is on July 17th, which is next Thursday. As you can see, 4:30 to 6:30, hybrid. Look it up, and I can certainly share the link, and you can get that. And more importantly, is September 11th. Come to Las Vegas, visit our beautiful event center. Dr. Lokshin just saw that a couple of weeks ago, and we will have 2 of my friends who are on this picture. That's Michael Okun that I was mentioning, and this is Ray Dorsey, who was a mentor of mine. He was a division director at Johns Hopkins when I was a junior, and so I took over from him, being a division director at Hopkins when he went back to Rochester. He recently left that. So this was their previous book, Ending Parkinson's Disease, and the Parkinson Plan. The Parkinson's Plan is being released. You can order it on Amazon right now if you want to. It's $30 for the soft cover. It will be shipped or starting to come out in stores on August 22nd. And we have this program at our event center for book signing, with those 2 authors being physically there and signing the books for you for free on September 11th in Las Vegas. So consider coming. It will be a fun event. We will have some educational program before the book signing as well. And these are my key takeaways. Thank you so much.
So we have time for a few questions. And for those on Zoom, if you can hear me, you can put the question in the Q&A box. And for those here in the room, we will repeat your questions so they're nice and loud. Yes?
Sir, do you happen to have surgical ability in Las Vegas?
Great question. So we have a DBS team, but we don't have Cleveland Clinic surgeons or hospital or inpatient anything. Right now we have a hybrid system in which we do the candidacy determination, we do the MRI, the videotape, do PDRS, on-off evaluation, the neuropsychological evaluation, all the required elements. And then main campus Cleveland Clinic has a big consensus conference where we participate, we present our cases, and then our patients will be flown to Cleveland, Ohio, get the surgery there, and we have a gift from Southwest Airlines that pays for the air travel. We have a patient gift that covers ground transportation, and then they come back and we do the programming and everything, and DBS care. We also have the ability to work with the local surgeon for those who don't want to travel to Cleveland, Ohio. Long term, we have some plan to be present in the north. So this may start with an office. We partnered with Barton Health, and so we will have an office building on Lake Tahoe. And if that works well, we do have, and I'm talking of years out, so it's not going to be this year. So we do have plans to have a Cleveland Clinic inpatient hospital as well. There, that's kind of more of a dream than plan. But the office space is a plan, because we already signed, they already struck the ground. So to answer the question, Briefly, we don't at the moment. We don't have a surgeon there. We have to find other ways to do the surgery itself. But in DBS therapy, the key is candidate selection. Because if you don't select the right candidate, it will not work. And we do the candidate selection. And two, management and programming is key. And so we do that too. So the surgeon, I don't want to in any way dilute the importance of their job, but that's a mechanical order. We tell them, do a bilateral STN implant, and they just do it. It's the neurology team that determines the candidacy and what target and what hardware.
Thank you. Other questions?
Do you have a phone number for the clinic in Las Vegas?
It should be on there.
Yes.
On the bottom of that flyer. If not, we'll make sure we get it out with everybody.
I don't see it here.
Yeah. I'll also give you a card.
We'll get that out
Thank
to
you.
everybody.
Yeah.
Absolutely. Another thing about research and involving some of the Revo Center is we did, as the Parkinson Support Center, actually advocated for a statewide research registry for all neurodegenerative disorders at the last legislative session back in 2023. And that will, things are slow, but it will happen, and that will be a patient-facing portal where you can learn more about brain health in general and see what studies are available for you to participate in if you are interested.
Absolutely, and I would add that clinicaltrials.gov allows a very helpful search by disease, by region, by study phase. So if you just put in your zip code, and you can extend it to 50 or 100 miles or whatever. And also, besides, again, clinicaltrials.gov, the other resource is on a Michael J. Fox Foundation's website. There is a trial finder, and that will not have everything like clinicaltrials.gov will have cancer and everything. The trial finder on the Michael J. Fox Foundation website, that it only relates to Parkinsonian disorders, Parkinson disease and Parkinson plus. And this slide, I just skipped quickly, but these are the studies that we are currently doing at the center. And you mentioned that we were the highest enroller in the tavapadone, and that's a big deal because that's a dopamine agonist that acts on the D1, D5 receptor subtypes. So it's technically a new in-class drug that is almost matching levodopa benefits in an early stage without the side effects and the baggage that normally associated with dopamine agonists. So it is a truly important drug. We are no longer enrolling, so I should not put it in there as active. We have patients who are still in the study and following through, but we don't newly enroll anybody. But we are newly enrolling in all the other ones.
Excellent. Thank you.
Thank you.
So please welcome Dr. Dagda.
Thank you, Dr. Lasky. And this is my second talk. I'm very honored to be here. And so for those of you who have seen my talk, I do have a few more updates, and I kind of mix it up a little bit more. So today I'm going to talk about some of the basic science research, and I know Dr. Mari talked about the importance of basic science and how this basic science was actually went to apply translational to apply science. And so my goal is also to give you a little snapshot of what I do at the University of Nevada, Reno School of Medicine, and how important, basic science and translational work it is to move the therapeutic pipeline for Parkinson's disease, especially in the current climate that we are with NIH funding that's being assaulted, literally. So as a researcher, we try to find different sources of funding at the moment, whether it's through foundation, NIH, or private. But This is a photo of my research laboratory in 2019. It just gives you an idea that, as Mindy mentioned, I train undergraduate students. My former graduate student is shown here. And so some of this work is highlighted in this talk. And also some of this work is highlighted by other people who contributed, such as Dr. Bridget Martinez, who did an internship and a fellowship at Reno, and Dr. Lombardi, who's actually helped with some of the immunological aspects of the work, and he is an associate professor in the Department of Microbiology. One of my former postdocs, Dr. Emmanuel Vasquez, who came from Mexico, did one of the two chapters of his PhD dissertation and got a lucrative job in Biorab in California, working for the industry. And Dr. Ain Gallagher, who recently got her MD, she also helped in some of this work that I'm going to show and highlight. Pretty much, she helped do some of the customer discovery data that I'm going to show. I want to start with the disclosure. Conflict of interest. I am a co-founder with Ruben Dagda for a company called CNS Curative Technologies. I'm also associate professor at the University of Nevada, Reno. And just want to highlight the sponsors, National Institutes of Health and National Science Foundation through the NSF I-Corps program. And so this is the main research building that my research lab is located at the Center for Molecular Medicine at the University of Nevada, Reno. And the company has an office space here at the Innovation Center. It's a great place to network and to have co-working space. It's a great hub to meet other people and network, as we're doing right now. And we meet other people who have startups who contribute to the entrepreneurial economy in Reno. So I know I'm preaching to the choir, but just want to highlight a few aspects about Parkinson's disease. And Dr. Mari made a great job to talk about Parkinson's, whether that's sporadic or genetic. I just want to mention, Parkinson's disease is one of the most prevalent neurodegenerative disorders. Just next to Alzheimer's disease, it affects 1% of the world population, as well as North Americans. And want to highlight that in about 60,000 Americans are diagnosed with Parkinson's each year. Here in Washoe County and Reno, there's been, it's very important to highlight that there's been a 40% increase in the incidence and mortality since 2017. So even though, other years, the incidence and mortality has been flatlined, this has definitely, the incidence and mortality has increased over the years, at least in Washoe County. It affects both genders, but mostly men, a ratio of 2 to 1, and it's caused by mainly the disease is characterized by motor symptoms, bradykinesia, rigidity, tremors, that starts with the arms and eventually progresses into the lower limbs, and eventually postural instability, which leads to imbalance and falls. One of the causes is the loss of dopamine neurons, which make the neurotransmitter dopamine, this gatekeeping, very important gatekeeping neurotransmitter that controls the rhythmicity, but also the neurotransmission of neurons. And it's treated with levodopa or Sinemet. But I just want to really highlight this slide. If you look at these cartoons, I know the motor symptoms decrease the quality of life, but it's very important to highlight that the non-motor symptoms as well causes a very debilitating decrease in quality of life. That can be constipation which precedes the motor symptoms by 10 years. That can be fatigue, which can lead into chronic fatigue. That can be loss of your ability to speak, the vocal cords, the energy to project your speech. And one of the most impairing ones is cognitive decline. That's basically what I want to show in this slide, is that it's very important that we want to, as scientists and also the medical field, develop new treatments to treat the non-motor symptoms. And I remember Dr. Trung had given a talk about non-motor symptoms, which was very good recently. So this slide just highlights that Parkinson's disease does not respect any socioeconomic boundaries, and celebrities like Muhammad Ali, Michael J. Fox, who has a great $300 million a year foundation that gives money for research and other causes. And you have Johnny Cash and Robin Williams, and who had a very severe form of Lewy body dementia, and his wife took over American Brain Foundation, and so also very important foundation to highlight. So Parkinson's disease affects every aspect, everyone in life, so it has no, it can penetrate every aspect of anyone in the socioeconomic barrier. And so this slide shows you that Parkinson's disease is mainly caused by loss of dopamine neurons that project from this very small structure, the substantia nigra, which has this dark pigmentation called neuromelanin. And those connections are lost between the midbrain and this large brain structure called the striatum, which is divided by putamen and caudate structures. So once those connections are lost, there's an imbalance and discoordination, because the neurotransmission going from the midbrain to the thalamus, into the cortex, into the spinal cord, all that communication is lost. And so one of the major hallmarks of the pathology of Parkinson's disease is the formation of Lewy bodies. This is an aggregation of protein trash that accumulates within neurons. So if you look at this color micrographs, you can see the Lewy bodies can take up to 40% of the space of one single neuron. This is the cell body of a neuron, and see how much of that is taken by this protein trash. And so eventually Lewy bodies will cause the neurons to die because they cause a lot of stress in the neurons, but that takes over 10 years. So there's this stage where the neurons adapt for a while, and Dr. Mari talked about the prodromal stage. And so, but eventually those neurons succumb and they die. But another aspect of the disease, besides looking at Lewy bodies, is that while 10% of the cases have a genetic nature, they're associated with mutations in over more than 30 genes so far. A lot of them, 90%, have no known cause. But the other aspect of this disease that's very important to highlight is that there's a loss of energy in the brain. And so for some reason, neurons lose their ability to make energy, and energy in neurons provide, that energy is essential to make neural connections, to make the neurons healthy. And the organelles that are contributors to make that energy in the neurons are called mitochondria. And so these are sausage-shaped organelles that make the molecule ATP, and ATP is really that powers your cells, and your cells powers your tissue. And so in the brain, we know in Parkinson's disease, there's a 30% decrease in ATP production because one of the main engines in mitochondria called complex 1 is reduced. It's somewhat shut down, but not completely. So this is what I really want to highlight, is that a loss of brain energy does contribute to not just fatigue, but the loss of those neurons. And so this slide shows that Parkinson's is multifactorial. Dr. Mari just mentioned, some of the causes are genetic, environmental. So you have toxins. You're exposed to toxins, and so there's an association of being exposed to toxins like rotenone, which is a known fish poison to control unwanted fish populations. People who have been exposed to this toxin have been associated with incidence of Parkinson's disease. Another one is MPTP, which leads to the formation of this metabolite MPP+, which is known in the 1980s. One of the neurologists found out that people who were taking heroin laced with MPTP develop Parkinson's disease. So that's a type of environmental exposures. And paraquat, another environmental contaminant, which is in the news most recently. It's been linked to Parkinson's disease as well as incidence of cancer and Agent Orange as well. One of the research papers that I published from my lab is that we also show, at least in an animal model, that's very intense psychological stress, whether, that can be associated with people losing their job or going through divorce. So in an animal model, we show that having rats exposed to cat urine, they kind of freak them out. They tend to develop motor symptoms of Parkinson's disease. And we know in some journals of psychology and psychiatry that psychological distress, being distressed and stress can be a risk for Parkinson's disease. Another contributor is, as you know, anyone who's over 65 years of age have an increased risk for developing Parkinson's disease. And we believe that one of the reasons is because as we age, our brain's ability to maintain healthy neurons, essentially removing some of the protein trash that I mentioned, like aggregated alpha-synuclein, which forms Lewy bodies, the ability of those neurons to remove the trash, called autophagy, this process of removal of trash, is decreased over time, and that leads to contributes to neurodegeneration. And also there's an increase in oxidative stress, so neurons tend to become, especially the dopamine neurons, become more stressed. And because they metabolize, convert dopamine all the time, that metabolic conversion of dopamine causes an increase in oxidative stress. So by and large, dopamine neurons are very sensitive to a lot of stress. But the best recourse to control motor symptoms is levodopa, L-Dopa, and this is a synthetic compound, and this helps to control motor symptoms. And as you know, one of the main problems is that Parkinson's patients lose their sensitivity to L-Dopa. It doesn't cross the blood-brain barrier very efficiently, only 1%, but it has a very therapeutic effect, which is 1% crossing the blood-brain barrier. And after 5 years of use, patients experience this on and off phases. And so to complement that, patients take dopamine agonists. Bromocriptine, bromorphin is one of those second line of therapeutics, and then you have the monoamine oxidase inhibitors type B, selegiline, rasagiline, to help reduce the metabolism of dopamine and increase the concentration of dopamine in the brain. As patients take L-Dopa, they get the second line of therapeutics to be complemented to help maintain that level of dopamine. So what do we need in the therapeutic pipeline, ideally, to treat Parkinson's disease? We want, a therapy or a compound that has a long-lasting effect, a single dose, not multiple doses, as with oral form of L-Dopa. Some patients take, L-Dopa orally 4 to 8 times a day. Single dose will be great. One that can complement existing standard of care therapy that can be disease modifying and reverse non-motor symptoms. And that can improve mood, cognitive abilities, reduce anxiety, and so on. So my research focuses on one specific type of compounds. They're called nootropics. They can be considered, these compounds are cognitive enhancers. They increase brain health. They can be also consider supplements. So nootropics can be both synthetics or natural. They can be derived from plants, or they can be synthesized in the laboratory. And so this is kind of a new, I will say, a trend in the practice, not just for Parkinson's, for several diseases. And I just want to mention that these are non-FDA approved for the most part. Some of them may be considered research grade, but they're prescribed by doctors. And so brain peptides, as you may hear in the recently, those are peptides that have been used not just for Parkinson's, but it's within this type. They're used prescribed by doctors for people who want to enhance muscle density and for exercise. But brain peptides are also cognitive enhancers to increase brain activity and cognitive function. And so within this, brain peptides fall within nootropics. And so I list some of them are included NoPep, which is the experimental compound that I use in the research lab to see how NoPep can improve Parkinson's symptoms in animal models. L-theanine, which is a compound from green tea to reduce anxiety. So it's an anxiolytic used in Parkinson's as well with caffeine. As you know, people who take caffeine have lower risk of developing Parkinson's. And so there's been a over 20 years study that actually shows lower risk of developing Parkinson's. Creatine, which enhances ATP production, mitochondrial function, can lower fatigue. And then forskolin is another experimental compound that I use in the lab. It actually increases this compound cyclic AMP in the body to increase dopamine signaling. And so one of the ones that most patients use are selegiline, even though it helps to reduce the metabolic inactivation of dopamine. Selegiline also has mild antioxidant neuroprotective effects, so it's somewhat considered within this field of nootropics as well. So selegiline is the one that's prescribed, FDA approved. The other one, modafinil, is a synthetic nootropic. It's actually prescribed by doctors to reduce cognitive decline. It's not a stimulant by any means. It's not addictive, but it does increase wakefulness and decreases narcolepsy in Parkinson's disease. And so that's one of the ones that has been prescribed recently by doctors to increase cognitive function and as well to increase wakefulness. So there's other over-the-counter dietary supplements that are actually prescribed or recommended by doctors to treat different symptoms of Parkinson's disease. And the market is still pretty nascent. It's new, unlike the market for Parkinson's therapeutics, which is within $5 billion a year. This one, it's barely breaking into $200 million a year. But it's increasing in trends. So what I want to actually show as part of my research is that we focus on two signaling pathways that are altered or dysfunctional in the brain, and that we actually made observations in animal models. So here I'm showing a slide on the left that a decrease in this very important neurotrophic factor, brain-derived neurotrophic factor, or BDNF, has been shown in post-mortem brain tissue. So if you look at some of the tissue that they look in Parkinson's disease patients who have died, there's a reduction of BDNF levels compared to control cases, and so about an 80% reduction has been observed. Whereas another area of the brain, the raphe nucleus, which makes serotonin, those levels are not affected. So this BDNF, it's very important neurotrophic factor, is involved in neuronal development. People with Parkinson's disease have been observed to have a reduction of about 50% of BDNF in their blood, and that's normally this reduction in BDNF is associated with mild to severe forms of depression, clinical depression. Another observation that we did in the research lab is that there's a decrease in protein kinase A. I know Dr. Mari talked about LER2, a kinase that's affected in Parkinson's disease, but in the research lab we found that in animal models that have Parkinson's disease, and this is in mice that have a mutation in this kinase called PINK1, they have a reduction in protein kinase A activity. So this protein kinase A helps to regulate BDNF levels. So when PKA is protein kinase A is active, it helps to increase the levels or regulate the levels of brain-derived neurotrophic factor in the brain. So when we actually made those and published those observations, me and my researchers, we came up with a way, maybe there's a way to normalize these two pathways, BDNF and PKA, in people with Parkinson's disease, and tested in animal models. And so we observed that, just want to make sure that there's a weight loss compound called forskolin and NOPEP. NOPEP activates AMPA receptors and helps to increase BDNF, and forskolin helps to increase cyclic AMP. It's an oral weight loss supplement that's currently being taken by people as a weight loss management. So we wanted to combine those two compounds to normalize this brain pathways. And so NOPEP has been developed by Russian scientists in the 1990s. It's actually used to treat Alzheimer's disease in Europe, and forskolin has been used in previous clinical studies to treat anxiety, to also help with weight loss. But since it increases cyclic AMP, it activates protein kinase A. We wanted to test those two compounds in the research lab. And so I know Dr. Mari talked about the importance of basic science. So we tested those compounds first in culture neurons. And you can see with a chemical called ronan, it's a toxin which destroys the dopamine neurons. You see the dopamine neurons stained with red. They disappear with ronan. But when we treated those dopamine neurons with both compounds, forskolin together with NOPEP, that were actually reversed, and those neurons were able to survive in the presence of this toxin. And so I just want to skip a lot of data to show that what happens in animal models. While it's interesting to see that the mice don't develop Parkinson's disease when we incorporate mutations, for instance, like PINK1 or LER2, they don't lose their dopamine neurons as much. The rats do, and so the PINK1 knockout rats came out about in 2017. I just want to show you a few data slides, not hitting you with a lot of data, but the rats do show degeneration. 50% of dopamine neurons are lost. They do show fatigue. They're tired. Their muscle strength and their back legs are weaker. They do show non-motor symptoms. They decrease ability to vocalize, which is similar, analogous to people with Parkinson's not having impaired speech. They show anxiety, and decreased motivation to move. And so, I just want to show you quickly that this is how we measure motor symptoms on a beam balance. This is 1 meter beam balance, and this is data that Dr. Grigorota, she's from Romania but living in Mexico at the time, and she did part of her doctoral studies in my lab. This is just a normal rat trying to move along a 1 meter beam balance. I know they take a little bit of time to move along, but eventually their tail is aligned in the middle and they make it to the end goal. One with Parkinson's disease, such as the one with a loss of PINK1, this very important kinase, they do have a little anxiety. You see they're wiggling their tail. It's not exactly in the center, and they take longer to reach the goal box. So they take longer, but they also, my experience, foot slips, and they might fall down. Eventually they struggle a little bit more. So I just want to show you that what happens in the same rat that we treated with forskolin. This is just injecting forskolin into the belly. It's called intraperitoneal treatment. This is before the treatment. You see the rat kind of falls down and struggling, having a lot of foot slips. Eventually fell down. The same one with forskolin. It really just whipped through the balance and actually improved. So it was, that time was kind of like a eureka moment for us. And we actually did different studies to show that treating rats, Parkinsonian rats with forskolin was able to reduce the loss of the dopamine neurons. And so if you look at some of this fluorescence photos, the red stain, it's kind of hard to see, but the more red dots, there's more dopamine neurons in the brain. The knockout has less of those red dots. With the forskolin by intraperitoneal treatment for 10 days, you see more of those red dots, and we quantify this. Each little dot is an animal. So with the P1 knockout, you see a 25 to 30% loss of those neurons. With forskolin treatment, we kind of recover that. So we also compare forskolin with levodopa, just to compare how it performs with standard care. And so the rats, if you look at the score, the higher the score, that means they have motor symptoms. And so this is the average score for falls and slips. The P1 knockout rats are shown in the red line that were treated with forskolin. There was a reduction of those motor symptoms. Whereas with levodopa, after 1 week of treatment, the rats responded with a decrease, but then they kind of relapse. And so it wasn't a long-lasting effect. With forskolin, you have this long-lasting effect in reducing motor symptoms. So that was like another eureka moment, but we wanted to actually develop this and actually apply this and translate it into from the bench side to the bedside. And so we thought about, we want to cross the blood-brain barrier, have these compounds reach the brain with more efficiency. We wanted to do this in an intranasal form. So we believed that at the time that we want to reach high bioavailability, so it's increased concentration in the brain, as a non-invasive method using a polypharmaceutical approach. That means combining different compounds, because there's no silver bullet to treat Parkinson's disease. So combine different natural compounds, no PEP and forskolin together, and other compounds to actually reach the blood-brain barrier and reach the dopamine neurons, the target. But also, since the cortex and other brain areas are affected in Parkinson's disease, we believe it'll benefit different areas of the brain as well. And so the idea is that taking a single dose will help to deliver those compounds into the striatum and into the midbrain. So that's the idea behind doing intranasal treatment. And so we tested this again in our animal model, the P1 knockout rats that I mentioned. And just to quickly run through the data, it did reverse the motor symptoms. It drove down the score of motor symptoms. Whether it's forskolin alone or the combination, it had the same effect. It reversed neurodegeneration the same way by delivering the compounds through the belly as intranasally as well. You see a complete recovery of the neurons, whereas the knockout, you lose about 30% of those neurons. And so each dot is an animal. So we look at least 30 animals per group. And also, it decreases the amount of protein trash. If you look at the little dots in the brain, those are what's alpha-synuclein aggregates, which is analogous to Lewy bodies. So in a normal age rat, you don't see those little dots. They're stained with green with the antibody. With the knockout, you see a whole bunch of little dots, which is suggesting there's an accumulation of this protein trash of alpha-synuclein. And with forskolin, there's a reduction, but with both compounds, there was an additive effect, and we quantify this. There's a reduction with forskolin in the number of dots. See, this is the mean number of dots. With the combination of compounds, it actually synergized the effect to completely remove this protein trash similar to wild type. So this just shows you that the combination of compounds that can actually increase brain protective pathways through intranasal administration may be a pharmacological way to treat Parkinson's disease, at least by suggested by preclinical data. I didn't have the slide to show you that one single dose had a similar effect for up to 5 weeks in the rats. And so this is almost, as Dr. Mari was saying, long-lasting can be disease-modifying. So 5 weeks in a rat, it equates to several months in a human being. So this is really promising. But before we jump from the bench to the bedside, I was encouraged to start a company to actually develop this formulation intranasally. And so the process involved asking different clients and customers, which are people with Parkinson's, to see what is it that they really want to see in a new therapeutic. And so this is a multiple iterative process. We went through this multiple times, and we actually, thanks to the funding of National Science Foundation, we carry out some of this customer discovery research for 6 months. And so we did a series of 110 interviews. Raul, who's here, we traveled the country from Sacramento to Phoenix to Nashville, Tennessee. Anya was doing New York. And so basically we went from 5 interviews to 95 interviews, asking patients what is it intranasal type of delivery of compounds, kind of like what you see with Inbrija, which is intranasal levodopa. Is this something that patients want to see? So we interview patients, companies, and influencers are the doctors and the healthcare practitioners. And this is what we learn. Patients are willing to pay $80 to $300 a month, up to $1,300 a month. For the other upside of the spectrum, they really want to treat their non-motor symptoms more than the motor symptoms, and that's understandable because the standard of care treats the motor symptoms. At the time, most patients were not taking Inbrija, 1 out of 29. Now I see that's 46%, so a lot of patients are comfortable with intranasal or inhalable forms of levodopa. But when we asked those questions, 90% were okay, and they thought this was a great non-invasive way because it will help to reduce the 14 plus doses they take orally at night and throughout the day, and this could be a way to treat their symptoms. And so going from the bench side to the bedside, I mentioned, we develop a company, and we have offices here, and my research, and this is our team, and I mentioned some of the members. A lot of the work has been supported by NIH, and recently one of the major grants is through Ascend 2.0 to use artificial intelligence to improve this formulation, to increase the solubility, the activity, and remove some of the damaging solvents that can affect the brain. And so we're using AI tools to do this kind of solubility and machine learning tool testing to improve the formulation. But this has been 10 years of work. And so the idea is that we want to develop something that's targeted, has a lot of brain bioavailability. I'm a pharmacologist, so I use those terms. And that can reverse both motor and non-motor symptoms. And so this is the pipeline. And so this one was launched. We're trying to get an FDA approval for Adapt Neuro, which is derived from the parent formulation of Neuroclimax. But I just want to show you the new stuff that came from 1 year of work. This is product performance. This is not clinical testing per se. This is, since it's over-the-counter supplement based on a formulation of natural compounds. This is self-reported data. And so we started asking before and after treatment, pre-post using the same customer discovery techniques that we learned of people who were taking this. This is just normal people. The combination of forskolin and no-pip seemed to improve on task, muscle strength, motivation, cognitive, and focus. This is, again, normal individuals, 23, not looking at Parkinson's yet. Self-reported evidence that they improve in energy, productivity, focus, and motivation, with 90% of clients report one benefit and at least 3 benefits in 60% of clients. Now, when we collaborate with a doctor and physical therapists in Greece and Mexico, we were able to look at some patients with Parkinson's disease. And so at least half of them reported a benefit, whether that's improvement in motor or non-motor symptoms, as observed by the physical therapies, and a little bit under 25% reported benefits. When we segregated both the people with and without Parkinson's disease, it seems that the people with Parkinson's disease responded a lot better. You see in the blue graph, bar graphs are higher than people that are normal. This is understandable because people with Parkinson's have a whole bunch of different symptoms, and problems with focus, energy, and productivity are one of them. And so they have a lower baseline of brain wellness as per se. This is our collaborator doing clinical observations in Greece. I don't have time to show the collaborators in Mexico, but he observed 30 patients per se and gave them this supplement. And what's interesting is 83% have better mobility. So he's a physical therapist, so he looks at movement, how much a patient walks in terms of meters. He even tested this in 81-year-old patients. 33% have improved. They have better writing or calligraphy for some of them, and 50% have improved energy. And just showing you his letter that he wrote saying that up to 10 patients with advanced Parkinson's disease, one with multiple system atrophy, which is a more advanced for worse and some with Lewy body dementia, had improvements in overall smooth sleep and overall energy. So this is where we are with the research. We want to actually move this to the clinic, test this in phase 1. And so I'm trying to get an approval for investigational new drug for this, for Adapt Neuro, which is based on the parent formulation of the compound. And I know we talk about exercise, and I want to emphasize not everything's corrected with drugs or pharmacology, but I'm 3 years into karate, and I like karate kempo. So I really encourage anyone who can do low impact exercise. I know in Las Vegas, we had a recent event that me and Raul went there, the Moving Day event. And so whether it's kickboxing or doing boxing, there's some organizations that actually help with that. People with Parkinson's, they have their own curriculum. And I actually discovered that my sensei graduated someone with Parkinson's. He was 56-year-old, so he's had some symptoms, but he got his black belt. And so I believe that exercise complemented with supplements and current standard care is the way to improve and ameliorate some of those symptoms, both non-motor and motor symptoms. So with that, I'll thank you very much. I have my contact, and if you have any questions, feel free to contact me or Raul, or if you have any questions right now. But thank you very much.
Thank you.
I went over.
We have some questions.
Oh, okay.
We have a question online on Zoom. How does this help people with dystonia?
Dystonia, we have not tested that. This is empirical data that is shown, so and it's self-reported. So that's something. It has only been observed with people with Parkinson's, some with multiple system atrophy, and some even with ALS, but we haven't looked into dystonia very specifically, but maybe our clinical collaborators can look into that.
Excellent. Questions in the room? No? Any other questions on Zoom? No. Well, I'd love to follow up on the exercise comment. Just so everybody knows, the Parkinson Support Center received a generous grant from the Reno Host Lions Club, and we offer a scholarship for people with financial need to be able to participate in Parkinson-specific exercise programs locally. And there is Tai Chi, there's yoga, there's rock steady boxing, and some other options that we're developing. So if anybody knows of somebody with financial need or wants to help contribute to that fund also, we do try to help people exercise and exercise with purpose, and in programs specifically geared towards those with Parkinson's. So just give a shout out to that. Well, thank you,
thank
Dr.
you.
Dagda. Thank you so much. So please welcome Nicole Tomitz.
All right. Can everybody hear me okay? Fantastic. Okay. I'll try to use my intent today when I'm talking, which is a word we're all going to learn here soon. But again, my name's Nicole Tomitz. I'm a speech-language pathologist and a doctoral student. I'm actually entering my 9th year as being an SLP, and I'm entering my 3rd year of my doctoral program. As mentioned, I am working under the guidance of Dr. Christine Galick. She sends all of her best wishes, but she could not be here today. As previously stated, my career has been focused primarily on swallowing disorders across a variety of diagnoses, as well as speech, which is, as the name says, a speech-language pathologist. This is often where I sort of get the question, why is a speech pathologist working with swallowing disorders? So I just want to emphasize that all of the muscles we use to talk were originally designed as the muscles we use to swallow. As we evolved as human beings, we use those muscles to swallow, and then we learn to talk. So that's where my role comes in. I'm very excited to just share with you today all the wonderful things that we have going at the University of Nevada Speech Pathology and Audiology Department, and how you or your loved ones could get involved if you would like to. All right. So the first thing I want to talk about is our collaboration with the Parkinson Voice Project. So in May of 2024, the Department of Speech Pathology received a grant from the Parkinson Voice Project to conduct research and provide free speech pathology services to people with Parkinson's disease for the next 5 years. So we are not billing any insurance, we're not even accepting it, we're not doing anything with it. Anyone who has a Parkinson's disease or a related disorder can come to our clinic and receive free speech pathology services. So the Parkinson Voice Project was founded in 2008. It is a nonprofit organization with a mission to help people with Parkinson's and related diseases regain and retain their speech and swallowing. They are currently on a mission to expand their philosophy to all 50 states. They're only in the 2nd year of this expansion, if you will. And so we've entered in their 2nd year, and I believe they're in now 42 or 45 states. I'm not sure you want to quote me on that, but we're almost to 50, so it's very exciting. And as that process, it was very competitive, so they only have 1 therapy and research center per state. So Reno got Nevadas. We beat out Vegas. What's up? So therefore, I would like to introduce that we have a no cost to the community clinic for anyone who wants to come. We are called the Nevada Speak Out Therapy and Research Center. And it's very exciting to not have to bill any insurance. So as part of our commitment to this grant, as well as giving out free services, we are also completing a research project. So this particular research project is looking at 2 things. I keep calling them tiers, so I'm going to stick with that. we are looking at the swallowing of people who have moderate Parkinson's disease. And so just a quick statistic, if you will, there's a little bit of bickering in the research as to how much swallowing affects people with Parkinson's. It's somewhere between 50 and 80% of people will at some point experience a problem swallowing with Parkinson's disease. So what I'm investigating as part of my dissertation work is these two tiers. So the first one is we're investigating the various components of the swallow mechanism across different liquid viscosities. And when I say liquid viscosities, I just mean if it's a thin liquid and progressively thicker. As humans, we don't just drink water. We like to drink water and juice and milkshakes, and sometimes we have soup. And so the thickness of the liquid varies as being people. That's what I'm talking about. And so we're specifically looking at moderate Parkinson's disease, and we're comparing them to people who do not have Parkinson's disease of the same age and the same sex. So as our first presentation kind of showed us today, when you're looking for a strong research study, you want to have a same age and sex matched peer to compare them to. That tends to make your science a little stronger. So that's the purpose of that. And then as you can see here, what we're doing is we're looking at them under video fluoroscopy. Some of you may have heard this term as a modified barium swallow study, if your doctor's ever brought that up to you. Modified barium swallow study and video fluoroscopy are interchangeable. So that's the first tier. We're just seeing, what does it even look like? So on your handout, you'll see that there's a couple questions I'm asking. So the first question is, what does the role of saliva play in drooling? For those of you who do have Parkinson's in the room, have you ever experienced some drooling that you perhaps would rather not have? Oh yeah, all right, there you go. So my first question is, does the volume of your saliva have any role in that? The second question I'm asking is, does your spontaneous swallowing rate affect your ability to manage your saliva? So again, perhaps you've had some drooling that you'd rather not. Is it that you don't swallow as much as you used to? Do you swallow more? That's the question I'm asking. Then I'm asking, is there an association between that spontaneous swallowing and the timing of your swallow? So we measure the timing of the swallow under X-ray. That's that modified barium swallow study. I'm also investigating, does the size of a drink you take affect your ability to swallow it? Again, there's some bickering in the research that says you should take small drinks, you should take big drinks. I would like to know what you normally take and what role that plays on your ability to swallow it. Then the second tier, if you will, is to answer the question, does speak out therapy change the swallow in any way after you receive the therapy? So I promise I'll get into what speak out therapy is, but speak out therapy has been known to improve the voice and the speech of people with Parkinson's disease. What we don't know is how specifically it affects the swallow. We know that people feel better, but we don't know why. And so that's the second tier of my investigation. So in order to do this, I'm trying to make it hopefully simple. So for the first tier, you come to an initial evaluation at the university clinic. We take all of those measurements I just told you about. And then on a second day, you'll complete that modified barium swallow study where we watch you swallow under X-ray. Then to answer the second question, so we have you come for 9 30-minute speak out individual sessions. It's usually 3 days a week for 3 weeks. So hopefully not too much of your time. And then we repeat all of those same measurements and that modified barium swallow study at the end again to see, did the therapy change anything? In what capacity did it change? So this is the point where I wanted to put some statistics on the screen, but I don't want to bias my study. So I will just say, We have 21 people currently enrolled in the study. Ultimately, we're looking to investigate 45. Of the 21 that we have initially investigated, we have found some statistically significant differences in the difference of the swallow between the person who has Parkinson's and the person who matched of the same age and sex. So it's very exciting. Already we've got some data we can report. But that one will probably have to be continued because I don't want to bias anything. All right, so if it were me in the audience at this point, I'd be like, okay, but what is speak out therapy? Let's talk about it. So perhaps some of you out there have heard from a spouse or a family member that you're a little harder to hear, or they're constantly asking you to repeat yourself. Yep, someone just said yep. Yep. So that's because what Parkinson's disease tends to do is it tends to make everything a little bit smaller, tends to shrink it. So that's why steps get smaller, the writing gets smaller, and then your voice will get smaller as well. That's why they're constantly asking you to repeat yourself. So the idea behind speak out therapy is that we're going to use a completely different part of our brain to activate more intention and more deliberation in the way that you speak and the way that you swallow. So the premise of the therapy is here on the screen. We call this the circle of intent. So as you can see, it's numbered and goes this way. So number 1 is a speak out evaluation. It's a really thorough evaluation where you come to our clinic and we'll take all those measurements I just talked about. You'll meet my graduate students. We'll take some other measurements of the way that you're talking, the rate at which you're talking, the volume and intensity of your voice. And then we decide if you want to do the therapy. If you do, we then provide you with a workbook. It looks like this. It's completely at no cost to you. And then also access to our e-library so that if you forget the workbook at home, you have no excuses. It's all online. And then we use that workbook in the individual therapy. But between that initial evaluation and providing you with everything you need, there is a What is Parkinson's video on the Parkinson Voice Project website. It's about an hour-long video where the founder of the program, her name is Samantha, she's amazing, where Samantha talks about how Speak Out was founded, the premises behind the program. So we usually ask you and your family to watch that together so everyone understands how you've now entered this circle of intent and what you're in for. And then we do those 9 individual Speak Out sessions. So they're 30 minutes, 3 days a week for 3 weeks, and then we remeasure. But the idea is that your therapy doesn't end there. You know, we just talked a lot about how exercise is kind of the key to everything we're doing. So your therapy doesn't end when those 9 sessions are over. What we do is we have you transition into one of our Speak Out groups. And so the Speak Out groups are created of people who have graduated from the Speak Out therapy. They meet once a week and we just do your exercises together as a group. And we kind of check in on how everyone's doing. And at that point, it allows us to keep an eye on everyone in case they need to do a refresher. So again, this is not, I'm not saying in 9 sessions I'm going to completely fix everything. I'm saying I'm giving you the tools to help improve your speech and your swallow. And so with those refreshers, every 3 to 6 months, you come back and you do another round of the 30-minute individual sessions, and then you go back to your group, and it just continues in that cycle. And as you can see, the complete around the circle is encompassed by that daily Speak Out home practice. So again, I'm not saying in 30 minutes with me that everything's going to be fixed. Again, I'm providing you with tools so that in those 30 minutes you can take it home, you can work with the workbook, the e-library, chat with your family, using more what we call intent and being deliberate in your speech. Okay, so that's a lot of talking. I would like to maybe show you an example. So I'm going to just show you a brief video of some before and afters of the Speak Out therapy. So I kind of threw it out to the room before, but I just want you to notice in the before video that perhaps this person is maybe a little quieter, maybe a little more challenging to understand. Look at her body language, and then just see if you can see any differences in the after. Oh, do I have volume? I hope so. I should. Yep, let me turn it up.
Go back to sharing yours.
Yep.
So we're not have technical difficulties.
Okay.
No sound.
Oh.
Now we won't.
We'll tell you what, I'll send it out, and then you can watch it that way. How about that? Okay, so I will make sure that everyone receives the PowerPoint, and then hopefully when you watch it at home, you'll notice, hey, that sounds different. Oh, am I not sharing? My apologies.
Yeah, that works.
Beautiful, okay. So a couple things I'll just point out, even though we didn't get to watch it. In the beginning video, and this person and I have discussed this in case she's watching, she felt that when she watched it back that she had a very flat sort of affect to her face. She felt that her personality had been stolen. She was very challenging to hear, and that she felt that she was mumbling. Her family was constantly telling her that she was difficult to understand. And then in the after video, we discussed she's much more animated. She feels like she got her personality back a little bit. She says that this is still a work in progress for her. But you can hear her, and the quality of her voice is stronger. And I keep looking at a couple folks in the audience who know exactly what I'm talking about, because perhaps they feel the same way. But I will send this out, and I hope you all can see the difference when you watch it. All right, so this is my call to action, if you will. So we are looking to enroll 45 people who have Parkinson's in this study. And we are also looking to enroll 45 people who are age and sex matched controls in the study. So there will be a total of 90 people enrolled in this. As I am a doctoral student, I spend most of my time trying to read as much as I can about Parkinson's, and so I can, impress the people on my doctoral committee and look like I know what I'm talking about. And I was reading 4 papers recently, and they actually only enrolled 10 people in each of these studies. And so the fact that we're looking at 45 with Parkinson's, I think speaks a lot to the robustness of how this study is going to come out, and I'm really excited about that. We ran what's called an a priori analysis to see how many people we needed to study to translate to the larger Parkinson's population, and that's how we came up with 45. So I currently have 21 people enrolled. We're looking for 24 more. So if anyone's looking to help us out, we would greatly appreciate it. And then of the same age and sex matched controls, we currently have 12, so we do need 33 more of those. That's really all I came up with today. I was sort of wanted to show you some cool opportunities we have going at the university, and maybe take any questions I could answer for anybody.
Thank you. Questions from the audience here in the room? Yes, in the back.
Yes. Hi.
Thank you. That was great. This is such an awesome service for people living with Parkinson's. I'm a prescriber. I'm a nurse practitioner, so I often refer patients to this and find that they decline after they leave speech therapy, and then we have to refer them again. So I love the cycle. It's amazing. Do you offer any of these visits virtually?
Yes, thank you for that. So we are asking that for the initial evaluation, folks come in person. I learn a lot about all of you when I can physically see you. But if travel is too difficult, all of the sessions can be completed via teletherapy. and then the groups as well. So we also do a virtual group once a week. So if anyone can't come in person to the group, there's always an option to do it over Zoom. Thank you for that. That's a great question. Any other questions? Yeah.
What is the curriculum entail?
Oh, great.
Intervention.
The actual intervention? Yes. So we do some vocal warm-ups to start off. And so those include, there's like a, they start with, we call them the may, me, my, mo, moos. So we go may, me, my, mo, moo. And then we do some sustained phonation, which is just where we hold ah at a good quality to warm up the voice. And then we also work on the range of the voice. So we do some glides up and down to help stretch out the vocal folds. And then we work on reading and speaking, so reading words or sentences, and then in conversation using that intention and being more deliberate about the way that we have people speak so that they're more understandable. So we do that in about 30 minutes, so it's very intense. But it's not too overwhelming. I'm hoping a few people can attest to. Great question. I saw one over here, maybe?
When will the study end?
Ideally, when I have 45 people enrolled. However, our goal is to use this free clinic that we've established to pursue other projects in the future. So this project will not die with my graduating as hopefully a doctor. I don't specifically have a date in mind, though. I'm hoping in the next year to graduate, but you know, I don't like to throw that out in case something goes wrong.
We have a couple questions on Zoom. One is, what type of changes did you see in the x-rays before and after, if that's something that you can say mid-study?
To be perfectly honest, I have not looked at the pre and the post swallow studies because 10 of these people are still enrolled, and I'm not trying to bias myself as an investigator by looking at what they did before the therapy, before they finished it. So I know that's not an answer, but
okay.
I can't share
And
that.
another question, you mentioned maintaining gains from the therapy. Can you speak more about that?
Yes. So the idea of the circle of intention is that once you've entered it and you've made some progress, ideally you'll continue to make even more progress, but ideally you also will not decline because we're keeping an eye on you. So if we notice that perhaps your voice is falling off, or if you're coming into the group complaining of new trouble swallowing, that's when we're immediately going to try to get you into a refresher. And we're going to spend more one-on-one individualized time with you to try to regain if you did lose anything, but hopefully maintain what you have gained. I do want to say too that this therapy, while it's fantastic, is not a one-stop shop, in my opinion. So if you complete this therapy and you need something even more specific that the entire region of speech pathology can offer you, other exercises, other interventions, those are still available to you through this clinic. I don't want you to think that I can only offer you one thing. I'm definitely here to make sure that anyone who comes to see us gets what they need.
And one more question.
Yes.
What is your contact information? And for those on Zoom, we will be sending it all out with slides and the handouts and contact information for all our speakers.
Yes, wonderful. Yeah, and for anyone here who got the flyer, the phone number for the clinic is at the bottom, as well as my email, and I'm available either way. So please feel free to call or email. Yeah.
Would you explain the scope of your practice for people that are not familiar with what speech-language pathologists do?
Could you repeat the question?
Yes, so the question was, what is the scope of what a speech-language pathologist does? Correct? Yes. Okay. So speech-language pathologists are responsible, my joke is it's everything from here up. So we're responsible for anything related to speech, swallowing, and cognition as well. So anyone who is having trouble speaking, whether that's forming the actual sounds, whether that's the quality of your voice, the formation of the words. We are also responsible, like I said, for the swallowing piece. So anyone who's having trouble manipulating food in their mouth, chewing it, getting it from your mouth to your throat, feeling like anything's sticking in your throat or like it's not going down the right way, or you're coughing or choking. And as well as from a cognitive standpoint, if you're having trouble focusing, any trouble with your memory, word finding, we can help. We have lots of interventions for all of those things. Thank you. Any other questions? Yes?
Do you have your clients use an app that measures the decibels of their speech?
That's a great question. Sometimes if people need that, we will use a decibel meter. Sometimes people can feel the difference in their body, and I do prefer people to feel that they're using intention and being more deliberate. However, as we know, biofeedback is sometimes a problem when it comes to Parkinson's. And so when you can visually see that you've made a change in your intensity of your voice, some people do benefit from that. So again, that's sort of an individualized practice, but yes, but no. Any other questions?
Any other questions on Zoom? No.
Thank you for having me. I really appreciate it.
Thank you so much. And that concludes our second mini research symposium. We hope you enjoyed. You had a full spectrum from full overview of how research is done to local products being developed to speech therapy available right here and now. So I hope you sign up for whatever you can participate in to learn as much as possible. Thank you.